Okay, so good afternoon, students. So, if you remember, we last time we were talking about uh, antigens, right? So we talked about the factors that makes an antigen good or a bad antigen. We learned few terms like what is immunogenicity, what is antigenicity, and immunogen, right? So we learned these terms, right? So I think now you can see the assignment in your uh, teams. Is it visible?
Yes, sir. Yes, sir.
So I think let's first start with that assignment MCQ. It's based on what we have learned in the last class. Okay, so it deals with the terms like immunogenicity, antigenicity, and the various factors that we learned last time. So once we do this MCQ, once we discuss the answers, then we will move on to the last factor which we had to discuss. Okay, so I think you can start attempting and then we can discuss the answers. So I hope everyone is able to do it. Okay, so I can see some students have started to submit. So rest of you start submitting maybe in next 2-3 minutes. So if anyone has joined late, then others are doing the assignment. So students, and many of you have started to submit. ये शक्ति,
Sir, I have a doubt.
हाँ पूछो.
Sir, I'm confused between like endogenous antigens and like the foreignness. If the antigen is endogenous, then how is it foreign? Like if we talk about cancer cells, like are they foreign? Um,
ये तुम कहाँ पढ़ रहे हो इसको? ऐसा तो कोई क्वेश्चन नहीं है.
no, sir, that's my question.
Okay, but I never read कि cancer cell is cancer antigen is endogenous, नहीं exogenous.
No, sir, it is endogenous, but will it be considered as foreign?
नहीं, तभी तो वो endogenous है ना, because
Okay,
it has.
so
Hmm.
okay, so endogenous
It is
antigens are not
not.
foreign.
It is not foreign, okay, but it's an antigen because our immune system has not seen it while uh, uh, we were developing. Okay, so there are a lot of such antigens which our body has not seen. Okay, so then it consider at non-self. ठीक है, बॉडी ने ड्यूरिंग द टाइम ऑफ डेवलपमेंट जो एंटीजेंस देखे होते हैं उसको वो सेल्फ मानती है. ठीक है, नाउ देर आर सम प्रोटीन्स, जस्ट फॉर एग्जांपल द स्पर्म्स, right? Sperms develop very late in life, right? And our body has not, our immune system has not seen the sperms, okay? So if because of some accident, some trauma, the sperms they get into our bloodstream, then there will be an immune response against it, right? Because it's, our immune system has not seen it, but it was always there inside us. Okay, similarly there are some proteins in the eyes which our immune system has not seen. Okay, so they are not foreign antigens, but they are um, antigens, right? The definition of antigen is that substance which can induce an immune response. Clear? But
Yes, sir. Thank you.
yes, when there is cancer, right, some new proteins are expressed on the surface of cells. So, uh, our immune system has not seen those new antigens, जो नए antigen बन गए या जो नए epitope बन गए on a cancer cell, right? So, body thinks कि yes, I have not seen this, so it's an antigen and I have to remove it. Okay, so then an immune response gets initiated. Okay, clear? Clear,
Yes, sir.
Shakti?
Yes, sir.
Okay, so students, I can see 16 students have submitted the response. So rest of you do it fast. Students, do it fast. Students, submit fast. So still, I think there are 2-3 students who have not submitted. So students, do it fast. Okay, so I think we can discuss the answers now. Okay. Now, the first question was a pathogen can be, right? So, an agent that cause disease, it could be a virus, it could be a bacteria, or it could be any agent. Okay. So, the correct answer is all of the above, and almost all of you, except one, has have given the right answer. Okay. So I think this term is clear. Pathogen. Pathogen is disease causing organism. Okay. A microbe may not be disease causing, but a pathogen is disease causing. Okay. Question 2 is the ability of an antigen to induce an immune response does not depend on the antigens which properties. So we are basically talking about the properties. So the correct answer is ability to enter the thyroid. Okay, so it doesn't matter if it enters the thyroid or not, but the ability of an antigen to induce an immune response will depend surely on its size, the dose, and the degree of foreignness. Okay, so almost again everyone except 2 students have given the right answer. Question 3 is antigens could be, okay, so they can be proteins, they can be carbohydrate, they could be nucleic acid, so all of the above. Okay, so again, all of you except 2 students have given the right answer. Question 4 was when a T cell, okay, so we are talking about T cells, interact with an antigen, what effector cells will be activated? Okay, and the answer is CD4 positive T helper cell, right? Remember, I talked about those thing when antigen interact with B cell, then what happens? And antigen when it interacts with the T cell, then what happens? So the effector cells in this case are the T cells. Okay, so they could be CD4 positive T cells, CD8 positive T cells, or any other. Okay. Question 5, greater the phylogenetic distance between 2 species, greater the structural difference, and greater the immunogenicity. The answer is true, and 1 student has given wrong answer, rest all of you have given the right answer. Okay. Greater the molecular size, lesser is the immunogenicity. The answer is false. Okay, 18 no 18 students have given the right answer. Okay, the rest of you have given the wrong answer. Question 7, more complexity of protein, more is immunogenicity. Answer is true, and all of you have given right answer here. And last question was, greater the heterogeneity, lesser the immunogenicity. So the answer is false, and only two of you have given the right answer. Okay, only two of you have given the wrong answer. Okay, so overall, I think the whole class have understood the factors which make like an antigen a good or a bad antigen, or what kind of immune response that antigen induces. Okay, so with this, now let me share the slide. So we were talking about the different factors, and in the previous class we skipped one factor because to explain that factor I need to explain you few more things. Okay, so let me share the slide. So students, do you have any question in mind before I start? Okay. Okay, so students, now talked about all these factors like foreignness, molecular size of the antigen, chemical composition and heterogeneity, and the complexity. Okay. Now another factor is susceptibility to antigen processing and presentation. Okay, so as I uh, discussed in the last class also, I talked very briefly. So what happens when a antigen, okay, enters our body? Suppose a pathogen has entered our body. Okay, so the antigen presenting cells, they will capture the pathogen. Okay, and once it has been captured, this will be broken down into smaller peptides. Okay, and these smaller peptides would then be presented to the immune system, where the T cells will come and bind to these peptides. Okay, now who present these peptides? It's the MHC molecule. So okay, so don't worry, we will be talking about MHC molecules in detail later on. Okay. Then uh, there are two types of MHC: MHC class 2 molecules and MHC class 1 molecule. Okay. And MHC class 2 molecule will present the cells to CD4 positive T helper cell. Okay, while MHC class 1 molecule will give it to cytotoxic T lymphocytes. Okay, so again, we will talk about this process in detail later. Okay, now macromolecules that cannot be degraded and presented with MHC molecules are poor immunogens. Okay, now what I am saying is, suppose an antigen enters our body. Okay, now the cells, when they capture it, they have to break it down into smaller peptides, right? Now just imagine what happens if this peptide is not getting degraded. Okay, the enzymes or the acidic environment, whatever environment the cell is giving it, that is not able to degrade this. So what will happen? This will not be seen by the immune system. Okay, so now degradative enzymes within antigen presenting cells can degrade L amino acids, but they cannot degrade the D amino acids, right? So now just imagine a pathogen which is having more of D amino acids, right? It will not be degraded. Therefore, proteins with D amino acids are poor immunogens. Okay. So just to explain you this whole process again, I will show you a video. Okay. So that will make things a bit more clear to you.
Today we will study processing and presentation of extracellular or exogenous antigen.
Okay, so students, in this video, we are just talking about the extracellular or exogenous antigens, which means the antigens which are coming from outside. Okay, and the whole pathway we will discuss later on, but this is just to make you understand or give you a brief glimpse of this pathway. Okay.
As the name indicates, extracellular or exogenous antigens are the antigens that originate from outside a host cell. For example, extracellular bacteria. These antigens are taken inside by cells either by endocytosis or phagocytosis. The cells involved in this pathway are antigen-presenting cells. Recall that antigen-presenting cells are phagocytic cells which present peptides derived from internalized pathogens. These include B cells, macrophages, and dendritic cells. The pathway starts when the extracellular antigens such as extracellular bacteria are internalized by the antigen-presenting cells. Let's zoom in to study this pathway. When an antigen-presenting cell internalizes a pathogen.
Okay, students, so you just saw, so this was the bacteria, this is the extracellular bacteria, okay, and this bacteria has got inside the vesicle, okay.
It internalizes it into an intracellular vesicle known as endosome. In the next step, this endosome fuses with lysosome to form endolysosome. In the endolysosome, the internalized pathogens are digested by proteolytic lysosomal enzymes into small peptide fragments.
Okay, so this is the important step which I was talking about, okay. So now what happens here, you can see here that your whole pathogen or bacteria, right, because of the presence of degradative enzymes and acidic condition, they have been broken down into smaller peptides, right? So the whole bacteria would not fit onto the MHC molecule, but these small peptides can now fit onto the MHC molecule, right? Now, as it was mentioned that L amino acids would be degraded, D amino acids would not be degraded. Okay, so in this case you can see it has degraded, which means they are good antigens, good immunogens. Okay, now just imagine if the conditions inside this vesicle did not allow breakdown of the pathogen, then it would have not generated an immune response, okay, or that would have been a poor immunogen. Okay, clear? So I was focusing on this point, and let's just now see what happens further.
MHC class II molecules are synthesized and assembled in rough endoplasmic reticulum. Here, the alpha and beta chains of the molecules associate with a third protein chain, called the invariant chain. This invariant chain binds to the groove of MHC class II molecule. This binding stops self-peptides in the rough endoplasmic reticulum from binding to the MHC molecule. These MHC2 invariant chain complexes are transported from the endoplasmic reticulum to Golgi apparatus and from Golgi apparatus to another vesicle. The invariant chain is digested and only a small fragment remain bound to the peptide binding groove of MHC2 molecule. This fragment is known as CLIP. CLIP stands for class II associated invariant chain polypeptide. In the next step, this vesicle containing MHC2 molecule bound to the CLIP fuses with the vesicle containing peptide fragments of internalized antigen. The peptide fragments displace the CLIP and bind to the peptide binding groove. These peptides are 11.
So students, now you can see that the peptides of that antigen, right? The smaller peptides, only they can get loaded onto this MHC molecule. Okay, so this is the most important point here, right?
11 to 15 amino acids long. The newly formed MHC2 peptide complex is transported to cell surface.
So now you can see that this cell is now showing this peptide to the immune system. Okay, so now the T cells which are outside, they will come and bind here. Okay, so all this is happening only because the pathogen or the antigen was degraded in the vesicle. Okay, तो वो डिग्रेड हुआ, इसीलिए ये सारा प्रोसेस हुआ. अगर वो डिग्रेड नहीं होता तो ये सारा प्रोसेस नहीं हो पाता और टी सेल्स एक्टिवेट नहीं हो पाते. ओके,
Once on the cell surface, they are presented to T cells. These T cells recognize the peptide displayed by MHC2 molecule by T cell receptor. Another receptor, known as CD4 coreceptor, binds to the beta-2 domain of the MHC2 molecule. Since this pathway involves MHC class II molecules, it is also known as MHC2.
okay. So let me stop this video. Okay, so is it clear? Okay, so you just saw the pathway, and what I am saying is macromolecules that cannot be degraded. Okay, so you just saw what happened. The bacteria came, it was captured, it was broken down into smaller peptide. Okay, and then those peptides were presented to the T cells. Okay, but the pathogens that cannot be degraded and presented with MHC molecule are poor immunogens. Okay, so is it clear?
Yes, sir.
Yes, sir. Yes.
Okay, so can anyone just explain me that process? Very simply. Anyone? Anyone wants to try? Okay, so I can see two hands, Manaswini and Shakti. So I'll start with Manaswini, and Shakti, you see it carefully, and later on if you find that some other thing needs to be added, you can add it, okay? So let's start with Manaswini. Yes, Manaswini. So you can also switch on your camera to explain us.
Okay, sir, 1 minute.
Do you need a whiteboard to explain this?
No, sir, I'll just say it through words. So
Okay.
basically, let's say once the antigen enters the cell, then there are other complexes within the cell which will recognize the antigen and will bind to the antigen. So these cells are called MHC1 and MHC2 cells. So they're the histocompatibility complex. So they bind to the antigen and they can engulf it through endocytosis, and they will slowly break it down into certain fragments. So once they break down the antigen, they take the fragments are basically attached to another part of the complex, which allows them to present the antigen to the other immune cells. And these immune cells will then come to the MHC complex, and they are able to combat the pathogen and ensure that the infection doesn't spread.
Okay, okay. So yes, Shakti, you want to add to it?
Am I audible?
Yes, Shakti, you are audible now.
What, sir?
You want to add anything to that?
No, sir. I actually wasn't able to hear her because of the network issues.
Okay. Okay, so I'll just try to explain this whole process again in very simple way, the way Manaswini explained. Okay, we are not going to get into details of this at this moment because you are going to read this whole pathway in detail later. Okay, so this is suppose your bacteria. Okay, and this is your APC. Okay, so let's understand what is APC. APC is basically antigen presenting cell. Okay, and they are three professional antigen presenting cells. So again, I'm not getting into details of APC. We will talk about APCs later. Okay, but there are three types. One is your B cell, one is your macrophages, and other is your dendritic cell. Okay, so there are three antigen presenting cells, and these antigen presenting cells, they express MHC class 2 molecule. Okay, so these cells, they express MHC class 2 molecules on their surface. Okay, so now what happens when this antigen enters our body? This APC recognizes it. Okay, and this is then captured. Okay, so what happens in, so when it is taken inside, it gets into a vesicle. Okay, so in this vesicle, this whole bacteria enters, and because of degradative enzymes, okay, and the acidic condition. So it goes through different vesicles. Again, I'm not discussing that because that I'm leaving for later. But this whole bacteria now inside is broken down into smaller peptides. Okay, and these peptides have to be very small so that they can get loaded onto MHC molecules. These MHC molecules are formed in the uh, endoplasmic reticulum. Okay, so this is your MHC molecule here. Okay, so what happens, uh, इसमें there is formation of Golgi, so there is formation of a vesicle here from the Golgi bodies, and ultimately uh, a vesicle is formed which is containing this MHC molecule. Okay, and then these two will fuse. Okay, okay, so now what happens, when they fuse, so it has now MHC molecule, okay, on which these peptides get loaded. Okay, so this is your MHC class 2 molecule because we are talking about antigen presenting cell. Okay, now then what happens, this vesicle, it moves towards the outer surface. This vesicle, it fuses with the external membrane, and this MHC class 2 molecule then gets expressed on the outer surface. Okay, and now it is showing this peptide to the immune system. Okay, and then if then it identifies, okay, uh, a T cell. Okay, and this T cell, through its receptor, it will bind here. Okay, and this will lead to activation of the T cells. So this is T helper cell. Clear? So I think this was the most simplest way to explain this. So I hope this is clear, and even if it is not clear at this moment, don't worry, because we'll be talking about this in much more details later. Okay? Okay, I think now we can take a break and have your attendance. So students, be ready with your cameras. Okay, so students, any question? Any question?
No, sir.
Sir.
Okay, let's have attendance. So, Adish,
Present, sir.
okay, Atoiba, Okay. Ayushi,
Present, sir.
Sam,
Present, sir.
okay. Devanshi,
Sir, she had a network issue.
okay. Ishita,
Present, sir.
Janvi,
Present, sir.
Kshitij,
Present, sir.
Manaswani,
Present, sir.
Namrata,
Present, sir.
okay. Nandini,
Present, sir.
yes, visible, audible. Nehal,
Present, sir. So my camera isn't working.
okay. Rupal,
Present.
Sahiba, साहिबा एक बार फिर से ट्राई करो. Sahiba, you are not audible, not visible. Okay, Sanjana,
Present, sir.
Shakti,
Present, sir.
Shivani,
Present, sir.
okay, Tanvi,
Present, sir.
Tridev,
Present, sir.
Vivek,
Present, sir.
Yukta,
Present, sir.
Yukta, you were not visible. Try again. Yuvraj, okay, Yuvraj. Okay, students. So, with these 5 points, we have discussed the contribution of the immunogen. Okay, so the factors that influence immunogenicity. So there were two factors, right? One is the immunogen, another one is the host. Okay, so let's also try to understand the role of host. Okay. So students, we have already seen this, the contribution of the immunogen. So we talked about foreignness, we talked about molecular size, we talked about chemical composition and heterogeneity. After this, we talked about susceptibility to antigen processing and presentation, right? And then we talked about the complexity of the antigen. Okay. Now, another is the contribution of the biological system or the host. Okay. Uh, Now, in this, the genetic constitution or the genotype of the host is very important. So, that decides the kind of immune response which will get generated. Okay. So, apart from genetic constitution, there can be many other factors, but one of the most important is the genetic constitution. Okay. So, the degree of response depends on the genetic constitution of an individual. Okay, and it all started with the work of Hugh David. Okay. Now what he did, he injected polypeptide immunogen in two different strains of mice. Okay. So immunogen was same, but he injected in two different strains of mice. Okay. So the mice had different genotype. Okay. Okay. So the scientist was Hugh McDavid. Okay. What he did, he injected polypeptide immunogen, okay, in two different strains of mice. Okay. Now these mice, they responded differently, with one showing high antibody titer and the other showed low antibody titers. Okay. So the immunogen is same. Okay. So अब immunogen का molecular size, chemical composition, heterogeneity, everything is same. Okay. But still one strain is showing high antibody titer, another one is showing low antibody titer. Why? Okay. So basically the difference is in their genes. Okay. The genotype is different. Okay. So McDavid suggested that the reason is confined to MHC genes. Okay. And that MHC gene product, they play central role in determining the degree of immune responsiveness. Okay. So at that time, right, so you can understand MHC का role क्या होता है, antigen को present करना. Okay. So in some mice with some kind of genotype, they are able to express, or they are able to present more antigen or the appropriate antigen, while another may not. Okay. So MHC is playing a very important role, right, in the degree of immune response that is generated. Okay. So initially it was thought that it all depends on MHC gene, but later on with a lot of other studies, it was observed that genes that codes for B cell receptors or the T cell receptors or various other proteins of the immune regulatory mechanism can also affect the kind of immune response. Okay. So basically genetic variability affects immunogenicity. Okay. So I'll just stop here because we have another class just after 5 minutes. Okay. So I'll stop here and we'll discuss this point in the next hour. Okay. So students, any question till this point?
No, sir.
No, sir.
No, sir.
Everything clear?
Yes.
Yes, sir.
Yes, sir.
Okay. So I'll just end the class now because we just have to meet after 5 minutes. So you also take a small break and we'll meet after a 5-minute break. Okay.
Okay.